Maybe it was off earlier. Okay.
This transmitter receiver is turned off.
Okay. So, good morning again. So, should we start? Okay, so today what I will do is, I will just extrinsic probes which are connected to biostructures or something like that. So this is something very interesting, but the actual philosophy itself is not very new, and then it was introduced in 1976 by these two guys, this and this guy, in a paper, and then along with Martin Karplus, these two guys also won Nobel Prize for in chemistry last year for proposing these multi-scale modeling methods where we can model complex chemical systems. So as I said, where do we need? Because which are the systems that we need these approaches? Why not? We can just otherwise we can just use some quantum mechanical approaches and calculate properties. Yeah, and then moreover, this enzymatic catalysis, it occurs in part of a protein, and then so to model all these systems, we need these hybrid QM approaches. So basically, we are interested in energetics of the system, or or the optical properties of these systems. So these are the cases that we need QM approaches like enzymatic catalysis, modeling, GFP protein, or extrinsic probes when bound to biostructures like DNA, membrane, or enzyme, whatever may be, very complex structure. So the philosophy is that, as I said, we are going to divide the whole system into two parts. The region of most relevance, which we are going to describe using QM approach, and then the remaining region we are going to use some coarse-grained modeling. So the idea is that then we need to define which is the QM system. So we should be aware of what we are interested in and where from the properties coming in. So we should have some chemical intuition to decide about this, because you can keep on including different residues, and then you can get then end up in very big QM size. So you have to check all these things. Then we also need to define the interaction terms. How this QM and MM they two interact. So basically we have QM energetics, MM energetics, and then these two interact. So due to that we have some additional contributions, and then you can model this additional contribution in different way, and then that we are going to discuss in some detail. So now we will discuss about how we can choose the QM model. Of course, if it is extrinsic probe, no problem. Your probe molecule is just QM region, and remaining environment will be treated as MM region. But then in GFP protein, it's not the case. You have chromophore, which is covalently connected to the protein backbone. So we can start with different models. You can start with the simplistic chromophore, and then you can increase. You can add groups. Actually, these groups come from the protein backbone. So you can keep adding the protein, the chromophore, or different groups to the chromophore, and then so you can build different models, and then you check, you calculate property whichever you are interested in for all these models, and then you see where it converges. For example, sometimes it converges for this size of the chromophore. Then you treat this as a QM region, and then remaining backbone you can treat as a MM region. So this is a chromophore in pH sensitive TGFP1 protein. And there will be always some difficult cases, and the plastocyanine is such a protein. It is a blue copper protein, and then the optical properties of this protein is due to the copper ion. So then we cannot just describe copper as the QM region, and then you don't get anything. So you need to include the residues, but then which residues? And then you see the structure is really very complex. So you have to carefully decide, like So you look for the coordination of this copper ion in the protein environment, and then look for the residues which are bonded to this copper ion. So you can see that it is in trigonal bipyramid coordination. So you can look for all these 5 residues, and then you can treat this as a QM region. Remaining will go into MM region. So as I said, you can describe these QM interactions in various ways. You can include van der Waals interaction between these 2 subsystems. You can include electrostatic interaction between these 2 subsystems. Also, you can include polarization interaction between these 2 subsystems. So depending upon what you include, we call it with a different name. So the first one is mechanical embedding. So this is the crudest model. Maybe we only get if we are only interested in the energetics, and if you want to remove some strain from the system, then this is all right. So we also need to have force field parameter for the QM region. So this is the QM Hamiltonian. So we just include the van der Waals term. You can see that some runs over all the molecular mechanics atom and then all the QM atoms. And this is electronic embedding scheme. So we have 2 cases. In one case, the QM region polarizes MM region polarizes the QM region, and then which is accounted using this 1 electron operator in the Hamiltonian. So this is the charge on MM region, the distance. So basically your QM region feels the electric field due to the MM environment in this case. But then you can also do this one, like you can just compute the interaction between QM and MM region using this formula. Like so, here we include the charges of QM region and then the charges of MM region. So there is an electrostatic sum, but then you need to compute the charges for QM region. So this is done on the fly. Like during the simulation, this is done. Then you use the charges to compute this term. So basically here the MM region doesn't polarize the QM region. So we also have the polarizable electronic embedding scheme. So what we do here is we describe the MM region using a polarizable force field. So we have to compute charges, multiple moments, polarizabilities for this MM region. So now the thing is that we also account for this one basically. So earlier in the electronic embedding we have this MM region polarizes the QM region, but then here we also have that QM region also polarizes the MM region because we have polarizabilities on each of these atomic sites of MM region. Actually, in fact, this is the one you are trying in your this application session. So you can also try, you just remove the polarizabilities, and then you can just try the electronic embedding, and then you can see like how much different are the results when you use electronic embedding or polarizable electronic embedding. So this also you can try. So the idea is that this polarization contribution, this is it is shown that not so important when it comes to modeling the structure. But for some cases, in the case of property modeling, this is shown to be significant. So we have to test. So what are the things can be done in the QMMM framework? We can get energies more accurately, and then this is very much relevant when it comes to drugs design because you are trying to find the interaction between the drug and the target. So even few kilocalories. Yes, please.
One question about the last type of embedding.
Yeah.
Because as far as I understand, in first 2 types of embeddings, you just add QMMM term as just extra correction.
Yes. No, actually, it goes into the SEF cycle.
Yes, but
Yeah.
it's an additive term.
No, in case of electronic embedding, no, it's not like that.
I mean, it's
Okay.
but in the last term, in the polarization,
Mm-hmm.
I think that instead of Schrödinger equation, you should use master equation or Lindblad equation because
Okay.
you have Hamiltonian principality in this term.
Mm-hmm.
because there is exchange of energy. It's not no longer isolated system. So in principle, it should be linear equation instead of Schrödinger equation. This is the first problem.
Mm-hmm.
And the second problem is that you don't know, in this case, the structure of the spectrum of Hamiltonian. Hamiltonian may be no longer.
No, Hamiltonian is known for the how to account for the polarization.
Maybe not such a joint in this case.
Mm-hmm.
So how do you treat this problem? Do you still use Schrödinger equation, or in fact you use?
So basically we use DFT. Okay. So and then only thing is we have this extra term in Hamiltonian, which accounts for the interaction between your QM and then MM charges in the charges in the MM region.
So
Or is it not clear? I can show.
in fact, we assume that this dissipative term does not influence the structure of Hamiltonian. Yeah.
Which one?
It doesn't change. But okay, so you treat it still as it is conservative, not dissipative.
I don't understand what you say. So this is the QM Hamiltonian. We are going to add this extra term, this one.
I think if there is a polarizability MM region, there should be some. We should enter the Hamiltonian by some Lagrangian multiplier or some, like you
So you still keep the same structure of Hamiltonian. You don't add some extra Lindblad generators or so, like in dissipative Hamiltonian.
I think
No, we added Hamilton extra terms, right? We added, or maybe I'm not.
Okay.
We can discuss, of course. Yeah. Yeah. So we are interested in energetics. Okay. So we can get more accurate energies than when compared to the other approaches like force field based methods. And this is very important, in these cases, like when you are interested in drug design or polymorphic prediction, you are always, dealing with in few calories difference may, change the results. So this is very important. So, and then this is available in Gaussian, and then it is called onium. Maybe I misspelled, but anyway. So then you can also model the structure basically. So what happens, how the environment is going to change the structure of the your QM region. This can be not only electron structure, but even the geometry of the QM region can be changed. So this is something relevant for these biomarkers that we are dealing with. So this is implemented in CPMD GROMAS, CPMD GROMAX. So then we can also compute the properties like linear, nonlinear optical properties. Yes. So the thing is that here you treat the whole environment explicitly. So that's a more important, interesting aspect. So this is implemented in Dalton 2013 actually. So this is also you are, in fact, you are trying this calculation of optical properties for PNA in different solvents. So yeah, so this is the summary. So the total energy, is the summation of QM, MM, and then QM, MM contributions, and then we have 3 different embedding schemes. So in this case, we only include the van der Waals, and here we include electrostatic interaction between these two subsystems. Here we also account for the polarization of the MM environment by the QM region. Yes. So that is the first part. Then. Okay. So now this is the second part, like where I'm going to discuss about what are the difficulties, practical difficulties when you want to model a biomarker. So then we are going to use some integrated approach, to resolve all these problems. Then I will show you some results from using this approach, some results from our own laboratory. So let me start with this, negative remark that the biomarker modeling is not so easy. Why? So I'm going to discuss about different aspects. So, so the binding sites for the biomarker or probe is the same. Within the biostructure that is, it's difficult to assign because sometimes you have just single binding site in this beta lactoglobulin protein, And then, for example, in DNA, we have 3 different binding sites: minor groove, major groove, then also intercalation site. Then in membrane, you really don't know where it binds, because you see, membrane is, you might have seen what is membrane, so you really don't know what is the binding site. So the binding site is not well defined in the case of membrane. So the binding sites, even finding the binding site even can be a difficult task. So now the other aspect is the probe, the biomarker, it can be extrinsic or intrinsic. It's in this case, it's like extrinsic, so it is not bound to the biostructure covalently. But then here in GFP protein, as we discussed, it's bound to the biostructure covalently. So now, as I said, the biostructure can influence the electronic structure of the probe, also the geometry of the probe. These molecules are known to change their geometry very much depending upon the environment. This particularly, this merocyanine we have studied extensively. We have seen that in a non-polar solvent, it is neutral-like, but then it goes to Studencka-like in more polar solvent. And then it also leads to changes in properties very much. Depending upon whether it is neutral-like or Studencka, the properties can be very different. In one case, if you look into the solitochromic shift, it is red shift. In other case, it will be blue shift. So this is something very important when it comes to the solitochromic molecules or biomarkers. So moreover, it's the salvation cell structure of this probe is changed by the biostructure. So which is this is like a short time scale process, and in addition to that, we also should have some methodology to account for the vibrational motion of the probe. Yeah, because in many cases the vibrational contributions are shown to be significant, like 5% to the total property. So then this is a long time scale process. The probe is very small, your biostructure is too big, but then even then the probe can bring changes into the biostructure. So this is of course a long time scale process. So in fact we have studied a case where the gold nanoparticle has changed the secondary structure of lysozyme. So a nanoparticle is like very small, but then lysozyme is relatively bigger size to protein. So these are all the different aspects when it comes to modeling the biomarkers. So we need to identify different binding sites for the probe within the biostructure. So basically we need to have some docking tool. So this we are not discussed, but this is not so difficult to learn. So then we need to model the small and long time scale processes. So we can use molecular dynamics or Monte Carlo methods. So then we need to account for the intramolecular motion of the probe. So for this we can use either ab initio EMD or corpuscular EMD. So then, as I said, the biostructure tunes the electronic and molecular structure of the probe, and that we have to use some QMMM approach. So then the properties, of course, you need to use DTDFT, and then we need to explicitly describe the MM environment. So you have to use DTDFT MM, so which is also called as QMMM response. So basically, we proposed in our laboratory this integrated approach. So you can see that you see all the tools, put together to attack this problem. So we have molecular docking here. So we look for various binding sites, and for each binding site we do this remaining procedure. So for the molecular dynamics, we get the charges for the probe from Gaussian calculation and then do the molecular dynamics in an explicit bioenvironment. Then we use the hybrid molecular dynamic simulation, as I said, to account for the small molecular motion of the probe, but in a more accurate way. But then, here you also need, when you want to do the TDDFT calculations, you also need the charges, polarizabilities, other multipoles. So then that you need to derive for the bioenvironment. So the force fields are derived in these steps. Like here you need for the probe, and then here you need for the region or your bioenvironment. Yeah, so it's like a very lengthy protocol we follow, but then we are able to account for different aspects. We are able to model or answer different aspects when it comes to the biomarkers and the protein complex system. So this I'm going to explain some of these aspects from our own studies. So how the probe binds to the target biostructure, and what's the driving force? Why the probe should go and bind to the target biostructure? So then in the vicinity of the biostructure, what happens to the probe's electronic structure, molecular structure, the dipole moment, and its geometry, bond length, everything. So we also analyze this aspect. Then we also look into the solvation cell structure, what happens to the probe's solvation cell structure when in presence of the bio environment. We also look into the properties, 1 and 2 photon optical properties of this probe in aqueous solution or when it is bound to some biostructure. So then finally we try to look for some structure property relationships so that which can be used to fine tune the properties later. So I can just look into these aspects in few of the cases, and then I will conclude this session. So first let's look into these binding modes of the probe within fibril, and we also look for what is the driving force for this probe to bind to membrane. So these two aspects we will see in this case. So this is the fibril, which is known to be the culprit in the Alzheimer's disease. So basically it's of interest to look for some biomarkers to find this. So this has motivated us to look into this aspect. So we have looked for different binding sites in this biostructure, fibril. So we have found 6 different binding sites, and then what we found is like this probe can bind to the biostructure either in the parallel or perpendicular fibril growth direction. So this is along the fibril growth direction, you can see. So but here it is perpendicular to the fibril growth direction. So then this is here we see like how this probe associates to membrane. So we start with some structure like this, and then we do long MD simulation. You can see that it goes into the membrane. So here I have plotted different energetics, that means subsystem energetics, the probe, membrane, but Coulombic interaction, probe, membrane, electrostatic interaction, then probe, water, electrostatic interaction, and probe, water, Lennard-Jones interaction or van der Waals interaction, you can say. So as you can see here, so the lower the value, this means it is contributing to the stability. You can see the more negative, then this means it is contributing more to the energy stability of this complex system. So when we start with, you can, maybe it's not clear, I don't know, but when we start with, you see the Coulombic, the block one, Coulombic term, that means between the probe and water is more, and then the other two terms like Coulombic and Lennard-Jones interaction between the probe and membrane was close to 0, because you see it's here at this moment when we start the simulation. But then as we proceed with the simulation, you can see the contribution from Lennard-Jones interaction between the probe and membrane becomes dominant. That is the blue curve. So what do we learn from here? So initially, the electrostatic interaction between water and the probe is dominating, but then when it goes into the membrane, it's the van der Waals and van der Waals interaction between the probe and membrane, which is the one dominating. So basically, so what we want to say that the driving force for this association process is the hydrophobic interaction between the probe and the membrane. So now we look into the, what happens to the molecular and electronic structure of the probe. So here we have looked into two cases. We'll look into two cases. So the first one, the Nile rat in the beta-lactoglobulin protein. So here this is from the CPMD trajectory. We averaged out the bond lengths, and then we looked into different parts like the donor acceptor region, and then the region which is not from the, which is now beyond, which is far from the donor acceptor region. So basically, we looked for this bond length and average of this bond length, then this bond length, and then these two bond lengths. So you can see that in the first two cases, there is not much change. Whether and then these three cases refer to, the first one refer to Nile rat in water. That means we do just a simulation only Nile rat in water, and then we also do the other two cases. We have protein. In the second case, it is on the surface of the protein, and then in the third case, it is within the protein cavity. So what we see here is, the bond lengths of this group doesn't change. Wherever the probe, whether it is in the water or whether it is in the protein, or whether it is in the protein's binding site, binding packet, there is no change in the geometry of this particular group. But if you see this one, CO group, you can see that in water, it is longer 1.28, and then it goes to 1.24 in the binding site of the protein. So basically, the bond length is larger in water solvent and then smaller in the hydrophobic cavity, or this in the site. So what it tells, this you can say that bond length increase. You can say that it tends to become like a Studencka-like. In this case, it is you can say that it's more like a neutral form, because CO it becomes C plus O minus when you increase the bond length. And then we also computed the dipole moment. You can see that in dipole moment for the Nile rat, you can see that it is 24 in case of water, and then within the binding site of the protein, it is only 14. So almost 2 order it increases when it goes from binding packet of the protein to the water environment. So, and then here we also looked into the charges. I told you to analyze the group charges for paranitronylene in different environment. So, and this has been done for this system here. So you can see the acceptor group charges is minus 0.5 in water solvent. It goes to minus 0.2, and then donor group charges, that also reduces when you go to the binding bucket of the protein. But the group charge for this group, it doesn't change much. You can see. So as I said, the biomarkers structure can be very different depending upon the environment, and then its dipole moments can be different, also the charges. So in fact, this is the reason why the property changes for this probe when you put it in water, or when it is in presence of some bioenvironment. Basically, this is the reason why the biomarkers, how the biomarkers work as a biomarker. So this is the thioflavin case. So we have looked into the dihedral angle, which tells the geometry of this phenyl, these groups, this phenyl and this 5-membered ring. How they are, what is the relative orientation? You can just compute the average dihedral angle, and then given this N-dimethyl group, how what is the relative orientation of this group with respect to this phenyl group. So we looked into different dihedral angles, and then what you find is that in water solvent it's more twisted, and then it becomes like planar when it binds to the fibril. So there is significant or important structural changes in these biomarkers. Yeah, so we also have looked into the optical property of this probe in different binding sites. So, so it's interesting that in some sites it shows red shift, and then in some cases, some sites it shows blue shift. Yes, now let's look into this, how the solvation cell structure changes when you have biostructure. So this we'll do for just the two cases. So this is a tyflavine tea, this is in water solvent, and then, but this is in different binding packets. I have shown you like 6 binding sites. Here I just only show for 3 of them. So you can see that the solvation cell structure can be drastically changed. So what is done by this? Like, so the number of solvents near the probe, it changes drastically depending upon the binding packet. So then this is also going to influence the properties. So you can see that this is the number of solvent molecules in the second solvation cell. Up to the second solvation cell, it's like 140 in the case of water. It can go. It has gone almost like by 50% reduced in case of different binding sites of the fibril. So this is the GFP1 protein chromophore in different forms. So you can see that this is the solvent density. It's quite different, particularly in case of anionic. You can see that there is a delocalized charge density. So the solvation structure can be very different depending upon which type of binding site or which type of form you have. So you cannot account for such effects unless you do such unless you use such integrated approaches. So this is very important. So now let's look into the properties, like how they change. So this was the one that I yesterday showed you. I asked you to try to get similar curve like this. So we also did this for a metal probe. You have metal, and then this is a probe actually. So what happens to the optical property when it binds to a metal? So the experimental data here, using different approaches, the experimental red shift was like 23, blue shift basically. The static models, they don't produce so well. But then you can see that this hybrid QMM-MD based approach for structure modeling could reproduce very well the shift when the probe binds to metal. What happens to the shift? It is very well reproduced by this approach. And this is the two photon properties. So this is experimental data. There are 6 times lowering of TPA cross section when this probe binds to magnesium ion. So the static model, you can see that it's no good. It doesn't do very well here. It almost gives a comparable number. And in fact, this was reported in a JAX paper. Then we wanted to test using this approach, and then in fact you can see that we have done fairly well in modeling this aspect, the decrease of TPA cross section when this probe binds to magnesium. So here also we tried the 3 different approaches: rigid MD, rigid molecular model for the probe, flexible molecular model for probe, and then QMM-MD for the probe. So the dynamic models are able to reproduce the magnesium ion induced decrease in the TPA cross section very well, but the static calculations fail. And so we attribute this is a dynamical effect that you need to account for to model this effect effectively. So this is the pH probe. So we have looked into the change in optical properties when you lower the pH. For example, this is the spectra. In a normal pH, and then this is in acidic pH basically. So I have compared to the experimental data. So there are 2 possible protonation sites. So either you can protonate here, or you can protonate here, or even here, I don't know. So but then you don't know which form is responsible for such shift. So what we found was that this was the form that was responsible for the, the experimentally reported redshift. So you can also get insight like which is the structure or which is the molecular form responsible for a different effect. So here in this case we say that this form is the one responsible for the acidic pH behavior of this particular probe. So then we also looked into the two photon properties. So we found that like with for the low pH structure there is in this case you can see that there is 3 times increase. So then we also used some two-state model to see like what is the reason why there is increase in the two photon absorption cross section with when you lower the pH. So you can see that the contribution comes from the change in dipole moment, and this is the one we get from two-state model, and then this is the one we get from the response theory. So you can look for different effects like which one is contributing to the property. And we also try to come up with some structure property relationship. So the spatial overlap and then two photon absorption cross sections. So this just tells you like it's related to intramolecular charge transfer. So lower the value, you have larger charge transfer, and then higher the value, it's the charge transfer is small. So you can see that there is a relationship. If you can increase the charge transfer, you can increase the property. You can increase the two photon absorption cross section. So and then this also clearly tells like why in the case of acidic pH the TPA value is larger. Yeah, so as you can see that it's possible, even though it is computationally very demanding, the biomarker design, but then if you put effort, it's doable. So that is a positive remark I want to make at the end. So thank you very much for listening. You have any questions?
Can you please go back a few slides? Was this dynamics for the case of the central molecule in its protein shell?
Yes. Because this is a pH sensitive protein. So
The protein shell doesn't want the water so much, right?
what do you say?
I mean, the protein shell doesn't.
That doesn't change.
Some water still binds.
No, water is also in.
What is this?
No, what I have shown is, I just only shown the chromophore and the water solvent density. So the protein backbone is removed, but it's there in the simulation. Protein backbone, this the protein is not shown here. Protein backbone is not shown here. What I have shown is just chromophore and then solvents in this case.
So the chromophore is there, the protein is there, and the water.
Yes. All of them are there, yes. But this is just to show you like how the solvent density can be different in different cases.
So this dynamics was.
This is just. This is it's with force field, yes. Force field MD, yes. As I said, this is like a long time scale process. The solvent has to come inside and then equilibrate. So then these results you should you should analyze using your MD trajectory because that accounts for long time scale. Yes. Yeah.
I have one practical question.
Mm-hmm.
Say that we have a molecule surrounded with some explicit water molecules in the first shell, and then such a complex we immerse in a box of classical water molecules.
Okay.
And during the simulation, I can imagine that these water molecules from the first shell can dissociate and then mix due to some entropic effect with the classical molecules. So can you somehow prevent?
What do you mean? So the.
I mean the explicit
Mm-hmm.
water can dissociate and then mix with some classical.
Yeah, this bond breaking. Dissociate means what? Bond breaking or just leaving the first cell? You want to say it's just leaves the first cell. Okay, so then your simulation breaks because you should have then bigger box. That within the QM box, your the atoms that you use for the atoms that are in the QM region, they should be within the QM box all the time. So if it goes to the second cell, then you have problem. It will break.
Actually, in
So, so what we do is like we usually just treat chromophore or only in the QM region. So we don't include explicit solvents because not needed because anyway there is dispersion interaction, the van der Waals interaction, electrostatic. When you do this corpuscular MD, you have all this. But if you include in the QM region, maybe dispersion is not there because probably you use BLEAP or some functional that is not accounted then.
Yeah, add maybe in practice, the Camshell software provides an option that you define the QM region. It starts from the at a sphere centered at the chromosome. So if this molecule diffuses away, then the program treated as a classical one. So it's not a fixed, this one is not a fixed as QM. It can be QM or ML depending where it is. So that's a practical way of it. I mean, if you want to do such simulations.
So basically you're saying that there is a software like that?
Camshell.
Camshell?
No, in CPMD it's not like that. Yeah.
Camshell is not free. It's just a interface, I should say, to correlate some discussion, for example, on the lambda for QM and ML.
Okay, so if you don't have any other questions, we can. Actually, Zinli wants to teach you about the Gromox MD simulation. If you are interested, you can listen to him.
I hope you still remember that on Wednesday we derived some force field for Gromax, so why not just continue and try to run some simulation with Gromax because it's also quite nice software from my experience. It's free, first of all, and it's also very well parallelized. You can have quite efficient, quite good efficiency running MD simulations on several hundred or even thousand processors. So I have again put all the necessary files in sharing Zinli directory. Here I have added one more step, step 6, and we stopped at step 4 last time. So there is, okay, I have updated this README file for step 5 and 6. So here at step 6, if you remember, we optimized the dihedral angle, force constant, for the rotation of this NO2 group. Okay. So what I want to add here is that in principle you can do the same for this NH2 group as well, but you know, nitrogen, this NO2 group sometimes can be tricky because it's not planar. I mean, not exactly planar. When you rotate it, this nitrogen bond loses its conjugation with this phenyl ring, and then this NO2 group become non-planar actually. So it will change from sp2 hybridization to sp3 hybridization. So this complicates the force field because you have already specified the angle, equilibrium, and force constant. So what I want to say is that the rotation, if you rotate this NH2 group, its motion is coupled with angles. So it's not only what you get from the QM energy contains contribution from both the dihedral angle and also the angle bending term. So it's you cannot separate it. So it's a bit tricky. But for this NO2 group, we don't have this problem. So it's better safe to, to leave these GAF parameters as it is. And we here show that in principle you can generate sort of more reliable if this rotation of the dihedral angle is sort of decoupled from the other degree of freedom. So it's just to let you know that it may not be very straightforward in some cases, and you need to be careful with that. So now we have optimized these 4 lines connected with the rotation of the NO2 group. Here we have 3.1, and then we go to the step 5. Okay. I give you access to all these files. So what we need is first some MDP files here. The first one for energy minimization, just to generate a reasonable starting structure. And the second one is for a production simulation, MDP. And of course, we will need the topology file, which we have optimized in step 4, and also the GUR file, which was generated in step 2 and not changed. So the first thing we do is like in Amber solvation. So we use this command, gembox. -CI means we insert one molecule. So of course it is our molecule in GUR format. And we insert only one, and we specify some box size in nanometer. So we remember that the length unit in GURMAX is in nanometer. So let's say usually the cutoff radius for the non-bonded interaction in atomistic simulation is around 1 nanometer. So we should have a box with its length longer than 2 nanometer, because we have this periodic boundary condition and mirror image convention. So let's say 2.2. But beware that this is much smaller than what you have in Amber. What you have in Amber is like you solve it the box with a margin of, let's say, 20. That means that from each side of the molecule, it has a distance of 20 angstrom to the edge of the box, which means that in sum, the total length of the box is over 40 angstrom. Okay, it's 4 point something nanometer. But for To speed up the calculation, I just, for example, to generate a small box. So what it did by this command is just you put this molecule into an empty box. The box has only one molecule and nothing else. So we run, okay. The output of this gembox command is out.grow. This is by default. You can specify your own name by the -O option. Okay. Now we leave it as out.grow, and we solve it by another gembox command. Use a different option. -CP means the box you are operating, and -CS means solve it. You can use some simulation box filled with some solvent molecules like a dichloromethane, and if you don't specify anything, by default it will solve it as box using water. Okay, so now it says it generated 340 water molecules. So in total there are 341 molecules. Let's check. Okay, it's quite straightforward to do this, I would say. Okay. Now I can show the box, draw PPC box. So now you see it's, now we have water around this solute molecule. And in VMD, if you like, you can check the periodic boundary. No, not for this one. Okay. Okay, you see, in fact, it's infinitely replicated in space in 3 dimensions, x, y, and I didn't show z. So also in this side, plus and minus. So it's infinite periodic boundary condition. Yes?
Does it make any difference that the molecule is the side of the box?
So it does.
It would be better if we put it explicitly at the center.
Yeah, I mean, visually it's better, but it doesn't make any difference in simulations.
Yeah, but I mean, from
Yeah.
the result.
Yeah. I mean, when you are making figures in your paper, so you should put it, more in the, how to say, in the center also. Yeah. But we have tools to do this, actually. I will show you later. So this is just an input structure. It doesn't matter. We have some tools to make our figure.
You mean that this molecule can be moved to the center?
Yeah. It can be centered. So the output of this gembox command is always named as out.grow, and the previous out.grow was renamed as like this. So you always have keep track on it. So if you check the output of this gembox, you will notice that there are 340 water molecules. That's what we are going to add in our topology file. Let's copy this one from the step 4. So first of all, we need So here, this atom types section defines all the atom types in your system. So these 6 atom types are for the p-nitroaniline, but nothing for water. So we need to add something. Water, oxygen, and hydrogen. Here I have a sample topology file where you can copy this. Just remember this is, let's see, this is comment line, tip 3p water. Okay. So here the second column could be either atom types or the nuclear charge of this element. It doesn't matter. In classical MD, the program ignores this column. So this mass you can either specify here or in each molecule you can specify here. So these are the sigma and epsilon Lennard-Jones parameters. So then at the end of after this PNA molecule, we should add the topology for the water molecule. We can also get this from. So here, these several lines represent a rigid tip 3p water molecule. See. Paste here. See, rigid water molecule. Water model. Okay. So the last few lines is system. System is just system name. Name of your system. Okay. And here it specifies what the system contains. So we have our molecule, and the number is 1. And then we have our water molecules, which is named as capital SOL. We have 340. So that's it. Then we can then copy this molecule top to system top. It's just my own habit to name this topology file as system.top. You can rename it whatever you like. So next we can run this grompp command. So we have our previous out.grow system.top. Yeah. So it did not complain anything. That means we have successfully generated this topo.tpr. Because we are just running energy minimization, which is a very, very small calculation. We can run it just by typing this command here. Here minus NT1 means that you are only requiring for one processor. That is, it's better to use one when you are running running your calculation, small calculation, by using this command line. Okay. So it's quite fast. And after 63 steps, it reached some criteria we defined. So it's acceptable. So the output structure is renamed as conf.grow. And then we copy this after energy minimization, like this. So now we have equilibrated energy minimized this system. And then we run the, yes? Hmm? Yes, I can show you. Mm-hmm. It doesn't look very different from what we generated. But energetically, it is lower. So it's better to start from this energy minimized structure, so that your MD simulation does not crash. So the next step is to run to grompp using this md.mdp, which is contain the options for production, for equilibration and the production simulations. -C. We use this Guru file after an energy minimization and also the same topology file. Okay. Then we have successfully generated another new topo.tpr file. And this is the input for your simulation. Since it is computational intensive, it's better to submit a job. So this is the command you will need. Okay. You will find this here, local. If you read the online manual of this supercomputer system, so I think it's here, yeah. So you can copy the script here and submit some job. I did this yesterday, so let's check how much time. Yeah, like 10 minutes on 8 cores, on 8 CPUs. So for now, you can actually copy the trajectory file from my directory to save some time. And you can try it out later by yourself. So this sub-GROMACS script will generate, let's say, since your input file is since your input file is named as topo.tpr, it will produce all the output file with the same name and different extensions. Here you can see a lot of output. Here, this is a log, or you can have a look at the end. It summarizes. Here you can see the time and the step and the bond angle, all the potential energy terms, and also temperature, pressure. By the way, this is an NPT simulation under normal pressure and normal temperature. So it's 298 Kelvin and 1 bar. Here is a summary of the parallelization and the time used in the simulation. So this one is the trajectory, topo.xtc. And you may notice that this is the largest file in this directory, which always is because the MD simulation produces big files. And this file is it's binary. It's not a text file. If you want to see what it is, you just cannot see. You cannot edit it by text editor like Vim or something. But we have tools to analyze this. For example, we could convert this trajectory using this command. This trajectory is a command of GROMACS. -F is specifies the XCC file, trajectory file, as input. And this is another input file, which is the TPR file we generated as the input file for MD simulation. And you specify your output file name is trajet.tgro, or any name you like. Here we specify that the precision is to the 4th digit of nanometer. That means if you use Armstrong, you have the 3 digits after decimal. And here PBC mole means we make the molecule whole. Since in simulation, a molecule could pass the boundary, then you have, say, 1 hydrogen of the water molecule in the other side. But we could always make it the whole to make better visualization. And here minus center, this is what you mentioned to center this molecule in the box. And since we produced the 2 nanometer trajectory, here we discard the first nanometer as equilibration. So minus B, 1000 picosecond. And minus dt means that we extract the snapshot every 10 picoseconds. So there will be 100 snapshot by this. By this command. Let's run it. So here you should select the group for centering. Of course, we want to center our molecule, which is index of 2. And then we select a group for output. But we need output for the whole system. It's 0, okay. So you see now it's working. So it's generated 100 snapshots. Now you can see that in every snapshot, the molecule is in the center. So in this case, it's good to study the solvation structure of the molecule, but you lose the motion of the molecule itself. So I will show you another way to. So you can use VMD to open some GUR file of your system, any file in principle. And you right-click here, load the data into molecule, and you choose your trajectory file, which is XTC file. So it automatically recognizes that GROMACS XTC complex trajectory, and you load it. Then you see the whole trajectory running, and the molecule moving, and rotating, and but it can take quite a while since we have 10,000 frames. Now it's loading like not yet 200. So you can try it later. Anyways.
This box is periodic, right?
Sorry?
This box was, the simulation was periodic.
Yes, of course, yeah.
Okay, and then when you extract some frames, you just move the molecule to the center, and it just rotates there.
Mm-hmm.
Yeah, okay, I see, because if it's periodic, you can move the whole structure.
Yeah.
It doesn't matter where to put it.
Yeah. But the original structure is important if you want to compute its diffusion. And at this time and at the later time, you have a distance, how long it has moved. But after centering the molecule, you don't know how much it moved. So that's what you lose.
Is it possible that the molecule touches the border of BBC, and then you have half of the molecule on like one side and the other half on the other side?
Yeah.
Again, doesn't make any difference.
No, there's no difference. But here.
The molecule can cross the.
Yeah, of course. This half.
Stay there at the border.
Let's say this it's a box. We can have half the molecule here and half the molecule here.
And it's again would not have any effect on properties.
No. Because we have periodic boundary condition. When you compute, let's say, the bond length, when one atom is here, one atom is here, then you see them very close, actually. After, the periodic boundary condition is applied according to the, you have it in the MC code as well.
Yeah. Because from what I know that, for instance, in periodic VFD, there could be some effect on this. I mean, at the border.
Yeah.
A bit different than exactly the same atom.
Yeah.
Same surround, but
Yeah, that may be possible. But in MD simulation, you have very simple algorithms, which I can tell you that there's no difference. So here you have this option when we generate our trajectory, which means that if a molecule crosses the boundary, we make it a hole so that it visualizes it better. You can make better figures. You don't have half molecule here and half molecule there. There's no difference, but you can always have a better figure. Yeah. So, any more questions? Okay. So that's it for step 5. Step 5. Because we generate a long trajectory of a MD simulation, but anyways, we will use only some of them, like 20, for example. For publication, you may need 100. So here, this 100 snapshots is contained in this trajectory.grow file. Then we go to step 6. It will be very, okay, I should first give you access to all files here. And yeah, I should remove these backup files. Okay. This is what you have in step 5 and step 6 here. I have already generated a lot of stuff, but in principle, you will only need this ProScript, which is already there, and this trajectory.grow file from a step file. So let's copy it from step file. Okay, all right. And you have this GenQMMPL. So maybe you want to have a look at it, but you can always check it later. It's used for just generate some Dalton input files. Here I have written that the usage is like you run this ProScript with option -QM16 means that your molecule has 16 atoms, or the QM region has 16 atoms. That is P natural aniline, and you specify the basis set for Dalton calculations. And note that this ProScript is just written for water, not for dichloromethane or any other solvent. Okay. So if you have some other solvents, it's better to write a new script in any language you like. So let's run this command. Progen QMM -QM16 minus basis. Okay. So it take a few seconds, and then that's it. You have a lot of. Let's take a random mo file to look at. I think you are already familiar with this format. Here is a bit different, where you can specify only 1.0 and 6, or you can use this format. So it's the same. And some potential file. Okay, it's the same as from yesterday. So Murugan, there's nothing at the end. So this format is correct.
Yeah, this
Yeah, yeah. Okay. So you can always compare this the mo file and the potential file and this directory with what you generated yesterday.
Could you give access to this directory?
Sorry?
Could you give us the access to this directory?
Yeah. Okay. Okay. I forget again. It's everything is yes. Because every file and the directory I created by default, it has no access. Okay, let's do this again. I didn't try this Dalton input files. So maybe you can try it. If it works, then it's good. If it doesn't work, you can try to compare what is wrong in these files. Okay. And of course, I just copied the same DAW file here. So the last thing that I want to say port files that contain all water molecules. Because I did not, the script does not cut a sphere of the water environment. Okay, so in this potential file, you actually have the cubic box, the whole box of water molecules. They are all there. So it might be better if you can cut the sphere of water molecules, which make it more isotropic. But I don't think there will be a big difference of the QMM calculation results, because the water molecules at the edge of the cubic is so far away, and they don't have much effect on the wave function or density function. So that's it. That's my part. Any questions, please? Yeah, if you don't have questions, then we are at our coffee break. And half an hour later, we start another application by Murugan. So, yeah, if you have any question, you can ask me later, always.
So, shall we start the application session? Are you ready? So actually, if any of you have the results, from this static and integrated approach based modeling, you can come and present. I can give you a chance. So you can come and explain like what results you have got from different approaches. So any of you have results? So you want me to explain something like something that which is not clear to you?
Which results do you mean? And Dalton ones?
What Dalton? Yes, Dalton results. Yes, you have some results. You can write some small script and then you can do the averaging of the first excitation, and then you can compare with the other models. So you have got results from static, and then now you have also results from dynamic, like 4 different approaches: rigid body, flexible body, then Carpenter MD, then the reparameterized force field MD. Because you have generated the input files already. Yeah, just to 5, 10 should be fine. Yes, ordinary averaging of see the lambda max average, and then you can compute the standard deviation excitation energy that you can use for the width. Yes, that you can use for specifying in the width for fitting. Of course, the amplitude, it will be again average oscillator strength. Yes. So if we can manage to get some results, we can see.